Today's speaker is Dr. Kedar Narayan from the National Institute of Health in Frederick, Maryland. He and his group have done some AI applications to segmentation that the core is interested in. As you probably know, we have a serial block-based SEM whose section quite a bit of data. One of the bottlenecks of that is doing the segmentation. So our hope is that over the next year, we can improve our segmentation capabilities significantly. So welcome, Kedar.
Thank you, Glenn. And thank you for hosting me. It's so nice being here. It's good meeting everybody. And I must say I was thoroughly underprepared for how warm it is today. I thought I'd be all fancy with this extra layer, and now absolutely sweating half of my body weight under this blazer. All right. Can everybody hear me in the back and in the front? All good? Okay. So my name is Kedar, and what I'm going to be doing today is talking to all of you about an approach that we and others have been developing in the recent past. It's called volume electron microscopy. And rather than jumping directly into the computational aspect of things, because I understand that the awareness about what volume EM is might be a little different within this audience. So I'll talk a little bit about some of the concepts of volume electron microscopy. I'm also going to talk about how it can be really empowered by using correlative approaches where you image the same thing using multiple imaging modalities. And finally, some computational work that we've been doing in the AI space to help alleviate bottlenecks in the pipeline. Okay, so I'll introduce volume EM. I'll give you a couple of examples from the wet lab side of things. Primarily with FibSem, I've taken out some of the other approaches for simplicity, and then finally I'll talk about images to insights with advances in AI. So overall, after this talk, folks should have a reasonable understanding of the positives and the negatives of volume electron microscopy, what to expect and what to be realistic about. You also have an idea of the kinds of insights you'll get from volume EM imaging, and then finally a little bit about machine learning and deep learning approaches for visualization and analysis. Okay, so what is volume electron microscopy anyway? The first thing I tell everybody is that it's not cryo-EM. So we did not win the Nobel Prize in 2017. That was cryo-EM, which is looking at the structure of individual proteins at angstrom level resolution. What volume EM is, it's the studies of three-dimensional ultrastructure of microns to millimeter-sized cell and tissue samples at nanoscale resolutions. So the thing that's unique about volume EM is that it's not a single approach. It's a collection of approaches, including electron tomography, focused ion beam scanning electron microscopy, serial block-based SEM, which you have right here with Glenn, array tomography, which I understand is also being offered at a different core, grid tape TEM, and finally X-ray microscopy. And if you plot a graph between XYZ or 3D resolution versus the size of features, you can see that each of these approaches has a sweet spot. And together they're called volume electron microscopy approaches. And so in our lab, what we do at the Center for Molecular Microscopy is we sit at the intersection of imaging, biology, and computation. And so what you're going to see is an example where these three expertise interplay and come together to get you biological insights. So to continue with what volume electron microscopy is anyway, with conventional scanning EM, you may be familiar with this. If you look at an SEM image of a lymphocyte, for example, this looks beautiful, right? You get these ruffles, it looks three-dimensional, but it is a 2D image. It's not a true three-dimensional image. And if you do transmission electron microscopy or TEM, you can see what's on the inside and on the outside. So SEM is just purely a surface phenomenon. Whereas TEM, you can see what's on the inside and the outside, but the problem is that you're imaging a very thin section of your specimen, right? Several hundred nanometers. Recent developments have allowed SEM resolutions to be almost as good as TEM, and the advantage, of course, is that instead of having a single 2D image, you can now get a stack of two-dimensional images. So this is also a T lymphocyte, except that instead of one 2D image, you now get a stack of 2D images. And the advantage of this is that this can now be converted into a three-dimensional reconstruction, right? And this is that same T cell, and this has just been false colored with the mitochondria in pink and the nucleus in purple. So the whole point is that you can get three-dimensional insights. And so I have, as I mentioned, there are several ways to do this. And what you're going to see here is a movie that we put together. It's just a cartoon of how to image cellular and subcellular features using SBF-SEM and FibSEM. And I'm happy to share these movies for your own talks later if you want. So these bubblegummy kind of things are adherent cells, for example, and you want to image them. The way it would work is you can fix them, stain them with heavy metals, and embed them in a resin, right? And that's what this block is. And then after you trim your block, the cells are right underneath that top surface. So in serial block phase SEM, in the tool that you have here, your scanning electron beam creates an image. Then you have a blade that sections through the sample in situ, and then you take the next image and you slice, and then you image and you slice and image and so on, and you get a 2D stack of images. But the same kind of feature, you can imagine that the sectioning instead of a mechanical blade can be done by this blue beam. That's the focused ion beam. And as it slices through the cell, the scanning electron beam can then take an image, and that's how you get a 2D stack of images. Either way, whether it's done by a blade or by a focused ion beam, you get that stack of images that can then be reconstituted into a 3D volume, and then hopefully you can get segmentation and analysis downstream. But there are a couple of other ways by which you can image features that might be on a larger scale, and this is where we would do this with array tomography and grid tape TEM. You sacrifice a little bit of Z resolution, but the advantage is here, just like in the previous movie, you have a whole field of adherent cells, but now I want to see all these cells. What you do now is, again, you fix it, you stain it, you embed it in resin, but now you trim your resin block to something that's about 1 millimeter across. And now you do your sectioning outside the microscope, and each of these sections is put on a wafer that's about 4 inches wide. And then you put it in the scanning electron microscope and just go scanning through the entire field until you finish the whole mosaic. And the other way of doing this is that you can take this whole field and put it on a conveyor belt, essentially, put that through a TEM, not an SEM, and that gives you your stack of 2D images of your huge field. Either way, that stack, again, needs to be computationally processed, and then you get a feel for what's in the cell, not just at a single cell level, but ideally at a multicellular level. So very quickly, that's a brief run-through of the various volume approaches that are out there and how they do things slightly differently and are better at slightly different things depending on what your question is. Okay, so volume electron microscopy is having a moment, right? There's been a lot of hardware and software advances that are sort of coming together, and along with the James Webb Space Telescope and CRISPR Anywhere, Volume EM was named as one of the 7 technologies to watch this year. If you want to read an exhaustive overview, a few of us wrote this in Nature Reviews that came out earlier this year. There's a couple of papers that have a slightly more practical description of how to generate volume EM data sets. And if you just want a very simple commentary, a few of us again wrote this in Nature Methods earlier this year that just talks about what volume EM is for a beginner. And it's if I might say so myself, it's a pretty interesting read. So volume EM publications are coming in thick and fast, right? You can clearly see there's this inflection point, right? There's a huge interest in the field, but there's a huge caveat, which is the y-axis is missing. It's on the order of hundreds of publications, whereas cryo-EM, which is much more established, got about a decade's worth of a head start. That y-axis would be in the thousands, and with electron microscopy, it would be in the tens of thousands, right? Of course, those techniques don't have this kind of inflection point, but just to tell you that volume EM is still in its nascency. The community is growing, so I'm deeply involved in building the volume EM community. And for those of you who want to get involved, please contact me. There's a great website, volumem.org. It's just all community driven, where you'll find many resources, including the Slack channel, email list, a set of publications, how to some basic training videos. We still need to do some work here, and some community resources. And here's where, Glenn, is your microscope on this map? You should do it. Please add yours, because we have a global map going of volume EM labs and core facilities, and it's a very nice way to get into contact with the rest of the volume EM community. So please, yeah, just join us. There's a lot to be learned and a lot to teach as well. Okay, I keep talking about, volume electron microscopy is great. You can view things in 3D. Why do we even care? The whole point, of course, is that if you have a three-dimensional object, you really should image it in 3D if possible. So if you take the simplest of all 3D objects, a cube, and if you take representative 2D images of it, depending on the angle and the location of your 2D section, you could get dramatically different 2D shapes that all correspond to the same cube. And so if that's true for just a simple structure like a cube, you can imagine how it would be for mitochondria. So all our textbooks suggest that mitochondria look like this, right? Kidney bean shaped things. This is a real mitochondrion in a cancer cell. So they can be profoundly different in shape. And you have to be able to image them at high resolution in 3D and in their entirety to really get a feel for what these organellar features are. The FibSem, I'm giving you an example of a volume EM approach here. This is a FibSem stack. This is what the real data set would look like. You're looking at the leading edge of a HeLa cell. This is the nuclear membrane. You can see mitochondria swimming in and out of the field of view, ER, and so on. It's a very small ROI, just to show you that it's not quite as high resolution as the TEM, right? It's close, but it's not the same, so it's slightly poorer. But the big advantage is that this exact same stack can be converted into a volume, which can then be sliced and diced in multiple ways. So the same data set, you can cut it in XZ and in YZ, and the resolution is exactly the same. That's because FibSem is an isotropic imaging modality. Those of you who have collected confocal images stack know that if you look at your data in XZ and YZ, your signals look like footballs, like ellipses. That's because the data in confocal, for example, is inherently anisotropic. Whereas in FibSem, it's isotropic, meaning that the data can be sliced and diced in any way without any warping. But like any other imaging approach, we're still stuck in this Bermuda Triangle. You can pick two, but not three. You have to get great resolution and a large field of view, but you're going to take a bucket load of time, or you have to compromise something else. Okay? So that's still an inherent limitation with volume EM. And the resolutions, as I mentioned, are slightly lower, and the biggest issue, of course, is that it is slow. So it's not like high throughput fluorescence imaging, whole plate scanning, et cetera, it's much better for deep characterization of features at the cellular and subcellular level. And a typical pipeline for volume electron microscopy is, say you're a core facility, right? Like what Glenn has. Your collaborator or customer sends you your biosample. So this is a biosample. You then do your sample preparation protocol to yield you your resin-embedded sample that's heavy metal stained. You then do your specimen preparation protocol, and the specimen is what actually goes into the microscope for imaging. Then you, of course, you get your raw data. You then do processing to get that nice stack or a volume. And of course, here's our big pain in the the segmentation protocol for you to do reconstruction and analysis. And in any of these cases, you can have light microscopy at any of these steps for correlative imaging. But the point is that volume EM pipelines are really complex and they vary by project. So what your question is will really determine how you go through this pipeline. And all of this is really powered by technology development. Those of you who have seen my talks before, this is the 5-pillar slide. These are the 5 technological pillars that our lab really focuses on in order to make a lot of these biological applications doable. Better sample prep protocols with fewer artifacts, efficient imaging by volume electron microscopy, because you need to get your data and graduate or get your grant, correlate and processing these data that come out of the microscope, segmenting and visualizing your features of interest, and finally, once you get all that data together, to be able to analyze the data, but also handle the large amounts of data that are going to be coming out of the microscope. So the overall aim, and this is our philosophy, is we are aiming to do efficient quantitative analysis of three-dimensional nanoscale reconstructions of tissue and cell architecture. In one sentence, that's the aim of our lab. I know there's many words, but really what it means is you're looking at tissues and cells in 3D at nanoscale resolutions, and the whole point is you want to eventually get to quantitative analysis, and you want to do it relatively quickly. So, volume EM is, as I mentioned, is the 3D imaging of cellular ultrastructure at microns to millimeter volume scales at tens of nanometers of resolution. It's a handful of approaches that complement each other and have favorite spots where they work really, really well. The whole point philosophically that powers us, our lab, is that traditionally electron microscopy has always been what we call search and snap mode, right? You're at the microscope, you're looking through the oculars, you go around, and you find something of interest, and you take a beautiful image, and there's your figure 1A. But volume electron microscopy, the way we want to do it is to automate large volume imaging. We don't want to be at the microscope when it's collecting data. And the magic really is before and after the imaging. And as I mentioned, volume EM is in the midst of an inflection, and we think you should join our community. So, before I get into the wet lab part of this talk and talk to you about some correlative work from our lab, I'll stop, take a sip of Diet Coke, and ask if there are questions. No questions. Good. This is either because the talk is great or everybody is having pizza-induced coma. But I'm going to go with option one. All right. So what I'm going to do now is give you a couple of examples of one relatively straightforward and one not so straightforward collaborations that we had in the recent past that use correlative FibSem. So how does this work? Sometimes you're not interested in the entire field of view. Sometimes you're only interested in a very specific region of interest. So how would you combine this, light microscopy and electron microscopy? Here's a little movie that'll show you one way by which you can do this. So this is using the volume EM approach called FibSem. Let's say you have a whole bunch of adherent cells on a surface, okay? And some subset of these adherent cells are fluorescently labeled. The point is that you image them on a gridded cover slip by your favorite fluorescent approach. You then fix, stain, and resin embed it just like previously. But the point now is that that etched gridded pattern is now transferred to the cover slip, to the resin, meaning that now you know exactly the zip code of where your already imaged cells are. So this is the resin surface at the bottom. Your cell is right underneath this pad. So in the FibSem instrument, you can put the you can protect the pad, and this blue beam that's coming down is the focused ion beam. And that basically acts like a nanoscale deli slicer, right? And it slices through the resin that contains the cell, and it exposes it in cross-section, and then the scanning electron beam comes and images it, and that's how you get image number one. Then the fib moves forward by like 5 nanometers, and you take another image, another 5 nanometers or 10 nanometers, take an image, and so on, and you get a stack of images that can be imaged at anywhere from 3 to 10 nanometers and have a Z section of anywhere from 5 to 15 nanometers Z. So it's very high in X, Y, and Z. And then the final step, as I mentioned previously, is segmentation. In this case, we're looking at a dendritic cell. And in 2D, you think they're dendrites, right? They look like spaghetti. But in 3D, it turns out they're not like spaghetti. They're actually like veils. So if you take sheets of lasagna and then look at them in cross-section, of course, they're going to look like spaghetti. But if you image them in their entirety, it turns out that HIV-infected dendritic cells can have sheet-like projections. So this is how you would do correlative FibSem. And so in one of our collaborations recently, a collaborator wanted to look at cell division in the male Drosophila spermatocyte, right? So the way this works is you have, here's the sperm cyst, by the way, these are the spermatids, but before they actually form the sperm in the Drosophila, which by the way are like a millimeter or so long, it's crazy that they can fit that into the body, but they do, they go through this phase called spermatocytes where very rapid and synchronized meiosis takes place. And that means that, and the question was, well, we know that certain lipids are required for correct cell division at this particular stage, but how do the lipids get incorporated into the plasma membrane exactly during cytokinesis? So the way this was done is the spermatocytes were dissected and then placed and then in a well, and then a drop of agarose was added. This was still difficult to do, but it was still, if you ask us, relatively straightforward. This is at the 16-cell stage, and when I say that this thing is fast and in synchrony, this is what it looks like. Watch. That whole thing constricting, it's myosin, and it basically is anytime a single cell divides into two, right? You've got to have that, the furrow, which basically pinches off the one cell into two. And this is done extremely rapidly and in sync. And the question was, well, we know that these specific lipids are required to be delivered here, but how does this happen? With light microscopy, you just don't have the resolution to do so. And so this is why we needed a volume EM. How is it that these guys are being delivered to the cell furrow? This was done by correlative microscopy, so these cells were imaged by LM, and then the exact same pair that you see here, which we captured exactly when the furrow was at its skinniest, that was then imaged by FibSem, and you can see immediately that this is much higher resolution, and you can see all the subcellular features, as well as these ill-defined gray blobs, if you will. And what we were able to do is to show that at higher resolution that specific labeled lipids were actually corresponding to the structures that I just showed you. So what does the 3D data look like? So this is the same data. This is just the furrow, and in green are all the unfused lipid compartments, and the blue ones are the ones that we happen to catch just as they're docked at the furrow. So that everything that's false colored is just segmented. Here's the actual data coming up. And you can see that these guys are docked right there, right by a proteinaceous collar, right around the cytoplasmic furrow. And so what this allowed us to do was to say, by the way, we followed this up with some TEM to show that each of these lipid compartments are in turn made up of very small granules of lipids, essentially allowing us to clarify this mechanism where lipids are endocytosed into these early endosomes. They form these intraluminal vesicles, which are what these are, and the intraluminal vesicles are the ones that go right to the furrow and spit out. Once they dock, they spit out the lipids, which helps provide the extra specialized lipids to cause the cytoplasmic furrow and cytokinesis to happen. So that's an example of how Clem FibSem worked. Another case to show you how painful it can get is this was another deep multi-year collaboration where we wanted to look at the dividing C. elegans embryo. So what you're looking at, this is in real time, not in real time, excuse me. This is in minutes. You can see at a single cell stage to a double cell stage, what happens is, I'll play that again, the male, the maternal and the paternal pronuclei, they're right here, they contain all the DNA, they come together, boom, the nuclear membrane dissolves, and once they dissolve, the DNA then gets separated out into the two daughter cells. And so the question Well, again, this is tightly choreographed, spatially and temporally. What you want to get is, once fertilization happens, you know that the maternal and the paternal DNA need to come together, right? So the pronuclei means that the DNA is already packaged in these structures. The pronuclei need to come together, but the nuclear envelope must dissolve in order for the maternal and the paternal DNA to mix, because that's how you get your first cell, and then of course that then divides into 2-cell, 4-cell, and so on. So the question is, how does this nuclear envelope breakdown happen? It must happen in a structured way, otherwise there's nothing preventing the DNA from just sort of spilling out and getting everywhere. So by fluorescence microscopy, you can tell that there is a fenestration or a gap in the stain. This is sp12 is a marker for the nuclear membrane and ER. And you can see the condensed DNA in red. And you can tell that when the maternal and paternal DNA line up at the equatorial plane, that they do so in a little bit of a gap. So there was this hint that there's that gap that does form, and of course it must form at some level, right? Because otherwise, how do you even get fertilization? But what does that gap really look like? Why is this so challenging? If you want to get the nature of that membrane structure, that intermediate, it's beyond light microscopy resolutions. Even with super resolution, you just can't see it. So you can't use LM. You've got to image it in their entirety in 3 dimensions. So traditional TEM, which is just 2D, can't be used. You must image them in their entirety. So you can't use TEM tomography, which is limited to about several hundred nanometers, whereas this is several microns. And finally, you have to image both the condensed chromosomes and the fenestrations, and you don't know in what orientation they might be. So other approaches are limited in Z resolution, whereas FibSem is not. So FibSem was the way to go. But there's another problem, is because this is so tightly regulated and so fast, even if you do get this beautiful embryo, you're hoping to see the nuclear envelope fenestration. But if you don't time it just right, you've just missed it, right? Telophase has already started. The chromosomes are already being pulled to either side. You've missed it by a few seconds. And because doing this by high pressure freezing is the way to do it, it's a real pain, it's difficult to do, and FibSem imaging also takes a long time. It means that you must get the timing just right in order to be able to get the DNA lined up at the metaphase plate just as the nuclear envelope breakdown is happening. So timing is of the essence, right? So you can't use fixed cells imaging, because it turns out that C. elegans embryo, they have a very, almost like a chicken egg shell, right? There's this really chitinaceous membrane, and so fixatives don't diffuse quickly enough. You can't do live cell imaging, because if you do live cell imaging, sure, you've seen your feature, but by the time you take it over to the EM sample prep, you've missed your time point. So, and you can't do high pressure freezing after the fact, because for the same reason, you can't find out where your embryo is. So the only way to do it is to do cryofluorescence microscopy, right? So you do high pressure freezing, which is just a way of rapidly fixing your embryo within the order of milliseconds, not minutes. So you know you fix it at a particular time. You image by fluorescence under liquid nitrogen conditions. You then bring it up slowly. You bring up the specimen slowly to room temperatures, do room temperature FibSem, and then you do some computational correlation, and that's how you get success. It only took us about 1.5 years to figure this out, but one slide is all it gets. And this is the whole worm. So this is a C. elegans worm imaged under cryogenic conditions by fluorescence. And H2B is a marker for DNA. sp12, as I mentioned, you can see where the cytoplasm is. So we're interested in these guys right here. This worm, after staining, after fixing heavy metal staining and resin embedding, is the same worm here. You can tell it's the same worm. And then this was sectioned, and that's what you see right here. And here are your embryos of interest. So how do you know they're the embryos of interest? You do something called correlation for relocation. And that means you've got your feature of interest that you imaged by LM, you've got your feature of interest that you want to image by EM, and you just want to make sure that you're imaging the actual same thing. So you do a simple overlay, and you can tell, okay, this is my 2D EM image, this is my 2D LM image. I know I'm sort of in the right area. This is where I need to collect my volume EM data set. And this was done manually. And then this area was then collected by FibSim. And so now you can see, here's the multi-cell embryo stage. Here are the individual cells, and you want to get exactly the phase where the maternal and paternal DNA are sort of parallel to each other and facing each other. And give it a second. There it is. Right there, you can see everything looks like just a single line because you've caught it just at the right time where the maternal and paternal DNA have mixed and they've lined up at the equatorial plane. Right? So this is the only way to do it, and we captured it in, and this process is done on the order of seconds. So we had to be exactly right. And now you computationally can correlate your features, right? Because you know this red guy is this guy over here, and so on. The final step is correlation for registration, which is much more exact. This is once you have your full LM data set and your full EM data set, you want to know what does this red blob correspond to. And we knew what this corresponded to. That's that guy right there. We knew that these two correspond to these two in telophase, and the question was, what is that, right? Theoretically, you should only get two. Why is there a third thing? And it turns out that if you go through the data set, you'll find there is actually this guy right here that's H2B positive, and it turns out that that is an autophagosome. And so all that extra DNA that's just, sorry, not DNA, all the extra H2B that's required for packing the DNA appears to be stored in H2B because you need all that H2B for subsequent cell divisions as well. So, so this is how this gets stored and then presumably recycled. So just to show you how you capture the moment of fertilization, this is what the actual volume EM data set looks like. Here's the maternal pronucleus, the paternal pronucleus, and here's the DNA lining up, and there's the gap. Right there is the fenestration, right? The gap in the nuclear membrane, which we absolutely captured, and this is what it looks like in 3D. I mean, it's been false colored and prettified by an artist, but this is the real data. It's not a cartoon. And the point is that you can see that the maternal and paternal DNA touch through these big fenestrations in the nuclear envelope. And because you can do this at different stages, because now you have the spatial and the temporal control, you can see that with earlier in the process, the fenestrations are multiple but small, and then they grow to be larger later in metaphase. And this is a little different as to what was hypothesized. The hypothesis previously in the field was that there was just one small nuclear pore that would just go larger and larger and larger and larger, which was certainly a valid hypothesis, but what we found out is actually you get multiple small fenestrations in the nuclear membrane, which then grow larger. Yeah, this is a novel three-way sheet membrane that we showed. So this is just an example of showing how this can be done by correlative clem. So what I am going to do is I'll stop now because I'm now going to switch over to the AI work, but just to make the point that correlation for relocation is different from correlation for registration. And so with correlative microscopy, there are many ways of combining the two modalities, so the question must dictate the approach. So I'll stop for a second before I move to the computation third of the talk. Any questions about? Yes, Jim.
If there's nobody else.
I was going to ask a question.
Please do, go ahead.
Well, I was going to ask about the Drosophila data. So you didn't need high pressure freezing so you have these sperm cells on PFA,
Yeah.
but then that's going to fix that cellular process. So
Yeah.
how does the timing work?
So the way we did this was with high pressure freezing. So we
Oh, so you did do high pressure freezing?
we could we didn't. In the data that I showed you, it was done by conventional fixation, and we just have the power of numbers. See, unlike the C. elegans, which is very, very difficult to get a massive number of cells to be perfectly fixed, with the Drosophila dissecting out multiple spermatocytes, and so our collaborator just put dozens of them, and then we just fix them with the agarose, and then only pick the ones that happen to be fixed at the right place. So we brute forced it a little bit. Yep.
So actually, it's a follow-up to your question. You said you also did a TEM
Yeah.
in that workflow.
Yes.
How? I mean,
Not of the exact same.
okay, so you again.
So in a spermatocyst, this was at the 16 cell stage, and so the great advantage of Fib, as you know, is that you can have extremely targeted imaging. So because they're very well synchronized, we imaged, perhaps you caught it, we targeted for FibSem a pair that was on the bottom right at about 4 o'clock. And once we did that imaging completely separately, you did the TM imaging, I think.
So
along the cells.
the 3D volume image didn't translate specifically exactly to the TEM. It happened to be in another location. You could grab it.
Correct. It
And
was
do TEM on it.
Which
is statistically probably just as
It is because we knew it was the same because we had the fluorescence live cell imaging of exactly the same spermatocyst. So we knew that all 16 cells were completely in sync. I showed you that movie, right? They all are in sync. So one of them was for volume EM and the other one was for TM because at the end of the day, gold standard for resolution is TM, and that's how we do it.
So am I allowed 2 questions, Kedar?
You are allowed 17 questions.
No, just 2. At least now. When you think about the workflow for volumetric 3D imaging, how much of that workflow includes also CLIM? In other words, if you were to say CLIM starts doing this on a regular basis, how many times do you think he's going to try to do correlative CLIM or would be requested to do correlative CLIM versus just, I got these,
Yeah.
you get my question?
Yeah.
Okay, how much? Or what's the
In,
ratio?
in our hands, just based on things that I would say, let me think this through. About half.
I don't know the answer, but I would have guessed also because there's a whole lot of rich data just available in 3D imaging, so I
Yeah.
don't
So
want.
a lot of times there isn't the need to have extreme spatial
Yeah.
or temporal control over the system. Sometimes you simply want to, I don't want, the phrase brute force to sound like it's lazy and dumb. But sometimes you just need to brute force a volume to just characterize what that looks like ultrastructurally. And you don't need light microscopy, or sometimes in tissue level imaging, you cannot get light microscopy imaging because how are you going to do it, right? You take something from, say, the mouse, and it's very difficult to get fluorescence imaging unless you permeabilize it and do expansion microscopy. And anytime you do that, you're destroying the ultrastructure and you can't get EM.
When you, I guess I did have a third one. When you do CLIM,
Yeah.
how much of it is to use this as a tool to aim the right location for the fib, and how much is truly correlative information? Do you
Yeah.
understand? I mean, in some ways, isn't it just an aiming tool? I know where to go now.
Yeah, so now that's exactly the difference between this and
Yes,
this.
yes.
So the majority of the collaborations that we have done are correlation for relocation. Just tell me, hey, I just use the light microscopy like a flashlight, right? Now I need to, now I know what zip code I need to go to.
Find the
to
needle
collect
in the
my volume.
haystack.
Needle in the haystack.
Got it.
But there are specific cases that we have done, like the Drosophila as an example, actually, where we needed, both the examples that I gave, as it happened, you had to have accurate 3D registration of the volume data set and of the light data set to be able to say that this LM image corresponds to a completely new or unexpected feature in the EM. See, because if you want to do correlation for relocation, you need to know exactly what your LM and your EM points to. And That's the only way you can overlay them. But if you're going for discovery, then you need to have fiducials overlay them computationally and accurately, and then you can find out what your unknown feature is. Okay, very cool. So this is a slide that I presented a few minutes ago. In the experimental pipeline, everybody knows this. Here's the bottleneck. Volume EM hardware and software has dramatically improved. The visualization and analysis is a real challenge for everybody, including, I believe, right here, right? Why is this? With fluorescence microscopy, you largely see what you're looking for. You want to see nuclei, you're staying with DAPI. You want to see mitochondria, you're staying with mitotracker. With electron microscopy, you capture the ultrastructure of everything, meaning that if you want to accurately and precisely extract your feature of interest from these really large 2D or 3D data set, this is very, very hard to do. It's important to do, and this is currently the rate-limiting step in volume EM, right? With fluorescence microscopy, you see what you get. With volume EM, you need to do something to see what's of interest. And this is the volume EM segmentation challenge. So there have been machine learning and deep learning based approaches, but polished segmentation is still slow, still required, and most often is manual. And so the problem, of course, is that people do this. This is difficult, this is exhausting, and error prone. And anytime I say something like this, I have to provide proof. And this is our intern a couple of months ago. So it really is exhausting, right? You have these data sets that can be generated in a couple of days, and sometimes they take months to segment out. And a back of the envelope calculation from our side has shown that it's something like 1.5% of data that is generated actually is segmented. And we think that we're amongst the better. In a lot of cases, there's glorious terabytes of data sets that are just sitting on a hard drive somewhere because there's nobody to segment it out. So how do we attack this, right? The only way, and I go back to the philosophy of our lab, right? We want to do efficient quantitative analyses. To transform volume electron microscopy, which is right now still qualitative, still representative observations, right? You get these images, you have asterisks and stars and arrowheads. To transform volume EM from qualitative representative observations to quantitative analyses, the only way to do that is automated segmentation of any feature of choice from any data set. And this is where AI comes in, right? So if I were to ask the humans in the audience, find the mitochondria in the image, all of you will immediately point at this guy and this guy, right? The humans would do it immediately. The AI would suck. It misses out certain things. These are called false negatives. It also erroneously adds certain things, and these are called false positives. So AI solutions are pretty bad. And the reason is that they don't generalize particularly well. What does generalization mean? So many of you may have heard of deep learning, and UNets are a favorite way of employing this. If you were to train a deep learning model, I should have had a slide on deep learning. Okay. The whole point of, there are multiple ways by which you can recognize features in an image. Humans do this even without thinking. We're extremely good pattern perceivers, right? And in fact, we often see patterns where there are no patterns, right? That's why illusions work, for example. So it turns out that training these kinds of networks to be able to detect these patterns in electron microscopy data or any data is pretty challenging. And the way you do it is you train these algorithms on massive amounts of data where you basically feed it pairs of images. You feed it the grayscale image, and you feed it what's called a ground truth. So you basically are telling the computer, the algorithm, hey, look, this is the image, and here are the mitochondria that should be on the image. And you do this for many, many images. I mean, tens of thousands, hundreds of thousands, and millions of images over and over and over. And finally, the neural network will get an idea of what a mitochondria should look like. But the problem is, this is how it's currently done. right? You acquire the data, you manually segment a subvolume, right? And then you train a neural network or model to recognize, say, mitochondria. And then you do something called inference. You run inference on the entire volume, right? So here's the amount of data that you've collected. You do the manual segmentation on a small fraction. You then train a neural network, and you say, okay, you figured out what the mitochondria is. Now find out where the mitochondria are in the entire volume. And it works. But the problem is, the minute you, let's say you image a HeLa cell, and you train a model to identify mitochondria in a HeLa cell. Now you take that same computer algorithm, right, the model we call it, infer it on a different data set, and it'll work terribly, right? So it won't recognize mitochondria in a muscle cell, for example. So our aim in our lab, going back a couple of years, several years, was a generalist or a universal deep learning model that can segment, and we're interested in mitochondria for a variety of reasons, that can segment mitochondria not just in one given image, but in any electron microscopy image. And why aren't we there yet? Our hypothesis was that during the training, and many people have done training of neural networks, the point is we hypothesized that we only train them in a narrow range of contexts. What is this like? Let's say your model is like some Joe Schmo on the street, right? We want it to be the Michael Phelps of mitochondrial segmentation. So instead of training some guy directly to become Michael Phelps, our whole idea was, why don't you show him what a swimming pool is first, and then start training? And this is what we call pre-training. And I won't get into the details, but I would urge for those of you who are interested to read this paper. Basically what we said was we collected every volume EM data set that's publicly available, 2 petabytes worth of data. We threw it at this model, and we didn't tell it to recognize any specific thing. We didn't say segment mitochondria or segment TR. We just threw it at it, and it's something called weakly supervised learning, just basically allowing the model to understand what EM or electron microscopy data is. Forget any segmentation tasks. Then we came back and we said, okay, now that you've figured out what electron microscopy data should look like, now let's give you a task. And the way we did that task was to give it the ground truth, right? This was mitochondrial labeling, and we had a huge number of interns who, over a period of time, taught the AI, the deep learning model, what mitochondria are. So we took, as I mentioned, something like 2 petabytes of data. We did some clever computations that I'm not going to get into. We then added to it 135,000 examples of unique mitochondria. And then we used all of this to train the data set. And these, by the way, these data sets are completely open source. They're available at Empire. I'll give you the link later on. But these are, if you ever want to pre-train your own model, feel free to use these data sets. And what comes out of this model is what we call MitoNet, right? It's a neural network that recognizes mitochondria, and we are very imaginative in our names, so we call it MitoNet. And it does very, very well, right? GT is the ground truth. This is what the mitochondria actually look like, and this is what the prediction is. And you can see that it's voxel perfect, but the model does make mistakes in merging and splitting. Sometimes if mitochondria are very close together, it thinks that 2 mitochondria are 1, and vice versa. But in terms of the actual shape, it does very, very well. And we also released benchmarks to prove that the model performs very well. And I'm happy to talk about the statistical details about this, but take my word for it right now. We beat the Mito-EM benchmark, which came out from Lichtman's universe, and the Heinrich et al., which comes from Genelia and Google. So it's the one time when I can say we beat Google. They'll probably destroy us in 3 months from now, but for now, our model beats theirs. So what is unique about this model is that every mitochondria is segmented uniquely and accurately. There's something called semantic segmentation, where all the mitochondria are segmented out. And that has its own utilities. But what we went for was instance segmentation. So in this data set, there are 15,000 mitochondria. I want each one of them to be mitochondria, or to be segmented separately, and that's what our model does. It can be done in 2D and 3D, and it is, well, I don't know about blazing fast, but it's very, very fast on your laptop. This was immediately after the paper came out, so perhaps a little excited about this. But it does work fast enough that you don't need a cluster. You can do it on your laptop. And this is what was on the, that came out on the cover of Cell Systems recently. So finally, one thing that we're very particular about is that deep learning models should be packaged into something that is useful for people. So one thing that I'm genuinely proud of is that the documentation is very good. We worked very hard on this, and in our lab, anything that's shipped out is always tested by high school interns. And so if they could install this, you can too. Please give Empanada a try. It stands for Electron Microscopy Predictive Annotation at any dimensions. But as I was talking to Glenn, we were hungry, it was late at night, so Empanada it is. And feel free to take a photograph of the QR code and give it a try. Let me show you how it works. It's a plugin on Napari. So Napari, like Fiji, is a software platform. The only difference being Napari is supported by Facebook or Meta, so presumably slightly better funded. And this is how it works. So I'm going to show you a movie of how to get Empanada to work. This is in real time, okay? So you open your file. This is the Napari window. You open your file. Here's your FibSem data set. Here's the nucleus, a bunch of lipids. And all of these are mitochondria. And this is a Z stack. And I want to segment out each and every mitochondria. I go to Napari, hit 2D inference, boom. This is in real time. I go to any other slice, hit 2D inference, boom. Each one of those mitochondria is completely separately segmented out. You can do this in 2D, and you can do this in 3D. So just to show you how powerful this is, on a single GPU, 16 gigabyte RAM, so this is not too powerful, 15,000 mitochondria in an hour and a half, or 61,000 in 3 hours. So you can get massive number of mitochondria essentially at one click. This is not manually cleaned up, so this is what the model output. And in contrast, it takes about a month to do about 200 mitochondria. So this is what Empanada can do. Please give it a shot. It will, and you can also create your own models. As I mentioned, if there are split and merge errors, you will find them, excuse me. You can just, we have a module that will allow you to just point and click and clean it up. So with that, I will just go over my overall conclusions, which is volume EM is pretty cool, and the bottleneck, which is segmentation, now has certain deep learning approaches that can allow for near universal or generalist models. There still is a lot of work that needs to be done, but at least this is a reasonable approach. And so this is allowing us to rediscover cell biology. There's been a lot of hypothesis-driven work in cell biology in the recent past, but just by looking at things in 3D and at high resolution, we're finding that we can rediscover whole things in cell biology. So I'd like to thank folks from Zeiss and Leidos and Frederick National Lab, NCI. These are folks in our lab who did a lot of the work, especially Ryan and Abhishek on the computational side, a bunch of our student annotators, and then Adam, Chris, Valentina, and Irene on the wet lab. Thank you. Yes?
Is the Empanada available on Windows or Mac?
Both. So it doesn't matter. You can, so Napari is a little more, the overall package of Napari, the install is slightly different, but once Napari is installed, the actual environment of Napari, of Empanada, equally good. Macs, Linux, and PC.
So we have it installed, Chase installed it. If you want to try it out on your data set, let us know.
Yeah.
So I was wondering, like, why you chose to do the instance segmentation, because clearly it worked, but like I in my head, when I understand, I would have thought that like it would have made generalization harder, because like it's trying to say like this mitochondria is its own class.
Yep.
And so then I would have thought like unless you get that exact same shape mitochondria.
Ah, good question. So the way it worked, I didn't get into details of this. The model is actually something called panoptic deep lab. So Panoptic Deep Lab, the way I'd like to think about it is, let's say you have 5 people walking on a beach. If you were to do a semantic segmentation, you would get sky, water, sand, humans. If you do instance segmentation, you'll get 5 humans. If you do panoptic segmentation, you get 5 humans and sky, water, sand. And so the Panoptic Deep Lab actually spits out both the semantic segmentation as well as the instances as well. So you aren't creating 61,000 classes. It's still one class of mitochondria, but it also spits out centroids of each instance in that semantic class. Okay, so it's still one class, but infinite number of instances of that class.
So it's sort of like an umbrella or like a nested.
It's you can think it actually happens in parallel, but
Okay.
but yeah.
And then sorry, I have
No,
another.
please,
question.
please, please.
So for wanting to segment something besides mitochondria, like neurons and neuron processes, like something that I was just messing around with a little bit, but having trouble with, is that if I get it more like a model more accurate for the cell bodies, then it messes up on the processes.
Yes.
Or vice
Yes.
versa. So like, do you have any
So
advice for that?
yes. So were you using Empanada or were you using something like Voss or some?
Oh, just when I was just trying out various AI models.
So the answer is likely because of context. So your cell body is like, it's like asking a model to get both a tree trunk as well as a twig. And so I would suggest that you create 2 different classes and then join the classes later on. So train one for your big cell body, say train one for your processes, and then you know you can just simple math, just add them later on. Just do an intersection over area.
And will Empanada work on like non-mitochondria then too?
Yes.
Okay.
So Empanada, we have used it for multilamellar bodies, for example. Other people have reported using it for cilia. Other people have used, there's some things for which it doesn't make sense, like ER, for example. There's no instance of ER in the cell, like everything is one ER, right? So that's not very good. Another thing that, because a lot of this was trained on a certain flavor of data, you have to be a little careful about the resolution of the image that you feed it. If it's too low or too high, it struggles a little bit. So for example, if your image was really high resolution and you've got these mega nuclei, and you say now segment out the nuclei, it will segment it out, but it will over-split it, right? So you'll have to do a lot of merge operations. So there are some things that we're tackling in version 2, which will be released sometime soon. Yeah, you first, and then I'll get to you. Yeah.
What would be the kind of, like, if I wanted to go, say, identify infected cells and decide whether their nuclei are larger or smaller?
Yeah.
What would be the kind of the time frame of, just being able to go say, I want to do a nuclei and that's it, to actually, and then get something to spit out? Is this like a matter of hours? Or
Yes.
like for an initial training, or is this take?
One mistake that we made when we released Empanada is we forgot to ask people to give their own models. Let's say somebody is interested in nuclei and has created a model. We didn't put a mechanism by which they could deposit that model, those model weights, back onto the Empanada website, which in retrospect was a big problem because somebody probably has a nuclear model that works just fine. It's just that we don't know about it. So, but the way you would do it is there is a module where you can create your own model. Typically, what we would do is it says pick n number of patches. That's left up to the user. It could be 32, it could be 64, any number, and you can then say choose those patches at random through the volume, or the user can choose them themselves. And then you get, let's say you get 64 patches. You now have 64 patches which you will have to ground truth, which will take you an hour, a couple of hours. And then you train the model, and that's going to take an overnight or so. But then you hit run, not just on this volume, but it should work on similar kinds of volumes for all nuclei. So that's how it would work.
So
Once you have that
let's just
nuclei
say
model,
initially from like, let's just say in a week, you could go from start to kind of
start to end.
finish
But the
useful.
biggest advantage of these is that these are ready to go models, meaning that if you get a good working model, you can use it not just for that data set, but just go on and on and on for all similar kinds of data, and similar kinds of tasks.
And then do you usually, if it's not working great, do you usually just give it more ground truths? Is that how you
Yes,
usually?
so there's a module called fine tuning. So let's say you have, and I had a movie, I should have included that. But yeah, if it does reasonably well but not good enough, you do something similar, which is you say, okay, here are 10 examples where the model failed. So you would specifically choose areas where it didn't do well, give it the corrected ground truth, hit it again, and we show repeatedly that that works.
Let's thank Kedar again.
One more question, sorry, it was the last one, or I can ask you afterwards.
He can, if you
You
have
wanna do
other
it afterwards?
questions, come up
Yeah, just come
afterwards.
to me afterwards, yeah.
Yeah, so we were not having lunch with the core. This is our last lunch with the core for the semester. And if you have further questions, like we said, come on up because we're after 1 o'clock. So, anyway, thanks for coming. We'll see you next semester.
Thank you.